Hi guys, as I said in our last recording, there are three major types of signal receptors: G protein linked receptors, also known as G protein coupled receptors or GPCR, tyrosine kinase receptors, and ion channel receptors. Today we're going to explore the G protein coupled receptors. A G protein linked receptor or G protein coupled receptor consists of a receptor protein associated with a G protein, hence the name G protein coupled receptor. Well, what's a G protein? A G protein is just a family of proteins involved in chemical signaling outside of the cell, which causes changes inside the cell. And it's going to involve GTP and GDP, which are kind of like cousins of ATP and ADP. Some examples of G proteins, G protein receptors are receptors for hormones, for neurotransmitters. These G protein coupled receptors are transmembrane proteins, meaning that they span the membrane. So part of the receptor is going to be in the extracellular fluid, spanning the plasma membrane and entering the cytosol. And the section that's in the cytosol is the part of the G protein coupled receptor that actually interacts with the G protein. So how does this G protein coupled receptor work? Well, here's the overall process. First of all, the receptor binds to a site on the outside of the cell, which then activates the receptor. So here we've got reception. Activating the receptor starts transduction. Now the G protein is activated, transduction is continuing. The G protein activates a cascade of further compounds. Again, transduction continuing. Usually that further compound is an enzyme somewhere downstream, and then it causes a change or some sort of response downstream in the cell. So this G protein kind of acts as an on-off switch. If GDP is bound, then the G protein is inactive. But if GTP or ATP is bound, then the protein is active. So here we see GDP bound to the G protein. And in this case, our enzyme, our target, is not active. However, when GTP is bound to the G protein, that activates it, thus activating the enzyme and giving us some sort of cellular response. Here's an elaboration on an activated G protein. So GDP gets displaced by GTP. What that does is activates the G protein, which then activates the enzyme, which is the target cell in this case, and that results in some sort of cellular response. Then we just return to the inactive form. In other words, GTP gets phosphorylated and GDP returns to the G protein, inactivating it. The response of a particular cell to the signal depends on the particular collection of receptor proteins or relay proteins and of the proteins needed to carry out a response. So what I'm saying is that one signal molecule doesn't necessarily always elicit the same response. There are multiple pathways that could occur because of the same ligand. It depends on the proteins, the relay proteins, and the proteins needed to carry out the response that determine what the overall pathway is going to be. The classic example of a G protein coupled receptor is epinephrine, also known as adrenaline. So adrenaline is produced in your adrenal gland, which sits on top of your kidney, and it elicits a variety of responses. For example, your heart beats faster, your lungs or your breathing becomes more rapid. But epinephrine or adrenaline is going to cause a variety of responses in your body. So let's take a detailed look at the action of epinephrine. So the first step is that the ligand binds to the G protein coupled receptor. That would be your reception. That G protein coupled receptor then undergoes a conformational change. It changes shape and it binds to the G protein, where the G protein is made up of 3 subunits, an alpha, beta, gamma, and it has a molecule of GDP. That's activating the receptor. So now we've started transduction because we've undergone a conformational change. That alpha subunit exchanges GDP for GTP, and the alpha subunit dissociates, binds to, and activates the target protein. Now, the target protein for epinephrine is adenylyl cyclase. That target protein, that adenylyl cyclase, can then relay a second messenger, cyclic AMP in this case, CAMP, and that second messenger continues the cascade of reactions. So, for example, it binds to protein kinase A, which then activates phosphorylase, which then converts glycogen to glucose. That is the response, converting glycogen to glucose. Or actually, the response is that you produce the enzyme that breaks down the glycogen to glucose. Then the GDP is hydrolyzed to GDP, inactivating the G protein. So let's take a look at this picture and annotate it. So here you've got the ligand. The ligand is what's going to bind to my receptor. In this case, it's a G protein coupled receptor. And the ligand in this case is epinephrine or adrenaline. I've got my G protein, my 3 subunit protein with GDP attached, and I've got my target protein, in this case adenylyl cyclase. So in the stimulated state, when epinephrine binds to my G protein coupled receptor, it undergoes a conformational change. GDP is exchanged for GTP. My alpha subunit dissociates from the G protein, activating my adenylyl cyclase, my target protein, thus setting off this cascade of reactions, which can lead to a variety of responses. Leads to increased heart rate, dilation of skeletal muscle blood vessels, and an enzyme that's going to break down glycogen to glucose. So this one ligand, epinephrine, can give us a multitude of different responses. So do you need to know the nitty-gritty, every single part of a G protein coupled receptor? No, this is what you really need to know. That a ligand binds to the receptor. So for example, epinephrine binds to your G protein coupled receptor. That G protein coupled receptor undergoes a conformational change, means it's changing shape, activating it. GDP is exchanged for GTP, activating the G protein, which then sets in motion a cascade of reaction, including second messenger, known as cyclic AMP or CAMP, that results in a cellular response. In this case, we specified looking at the pathway of adrenaline that would cause an enzyme to be released that breaks down glycogen into glucose to give us a little boost of energy when epinephrine is released. As we noted before, the G protein system cycles between on and off when the G protein linked receptor is activated by binding with an extracellular signal molecule. The receptor binds to an inactive G protein membrane, which leads the G protein to substitute GTP for GDP. That G protein then binds with another membrane protein, often an enzyme known as the target protein, altering its activity and then leading to a cellular response. So here again is just another depiction of this same G protein coupled receptor. These G protein receptor systems are widespread, they're diverse in their functions, and they seem to have evolved very early. Several human diseases, including bacterial infections, interfere with G protein function. So why should we care? Well, because your vision, your smell senses use these G protein coupled receptors. Like I mentioned, diseases such as botulism, whooping cough, and cholera produce toxins that interfere with these G protein coupled receptors. And because of that, almost 60% of our medication works by affecting those G protein coupled receptors. Furthermore, a lot of drugs, including heroin, work that same way. The next type of receptor protein that we're going to discuss includes the ligand insulin and tyrosine kinase receptors.